Okay, we're back for urinary system number 2. We're going to talk a little bit more about the functional purpose of the kidney in regards to the biggest thing about the urine formation. It's not an easy thing. It takes a little bit of thought process to be able to figure this out, but it's very unique the way it happens. So let's just delve into it. We should know what all the organ structures are, so that should be one thing. But now we're going to go into the nitty-gritty and we're going to get small. I mentioned before that the functional unit of the kidney is called the nephron. Okay, and the nephron's where all the magic happens. In the kidneys there are millions of these little nephrons. Okay, they really start at the glomerular area, which you see is that little ball in the middle. It starts there, right around the junction between the renal cortex and the medulla. And a lot of these tubules will extend into the medulla, and definitely the one on the left that looks like the big horn, which is the collecting duct or the collecting tubule, does go all the way down through the medulla and empties eventually into one of those calyces, like we talked about before. The place where this all starts is at the area that what's called the renal corpuscle, and there's the renal corpuscle right there. The renal corpuscle is that group, that cluster of capillaries, that it's a ball of glomerular capillaries. Okay, has an afferent arteriole that goes in, like we talked about at the very end of the last presentation. It goes in, and an efferent arteriole that comes out. Again, that's the unique vascular structure that we find of the nephron. We have an arteriole, a capillary arteriole, and more capillaries. Okay, so this area, the renal corpuscle, is where filtration occurs. Again, we talked about how the efferent arteriole, the one leaving the glomerular capillaries, is a little bit smaller than the afferent arteriole, the one that's going in, so that increases a certain amount of pressure inside the glomerular capillaries to allow them to filter the fluid. Okay, around that ball of glomerular capillaries, there's a cup, like I mentioned briefly last time, and that cup is called Bowman's capsule, and it's basically a double-walled epithelial capsule around the glomerulus to be able to catch whatever is filtered at the area of the glomerulus. So basically, that Bowman's capsule will actually catch everything that's filtered in the glomerulus. So that's what we have there. It's the renal corpuscle. What happens is once the urine is made, okay, and let me just try to draw a little picture here. Once the urine is, or I can't say even urine is made, once we have the filtration that occurs in the area of the glomerular capillaries, at that point what happens is the filtrate starts to go out through this area right here. It goes through this tubule here, okay, loops around like that, okay, and that's called the proximal convoluted tubule. So let's look at that. So once the filtrate occurs at the area of Bowman's capsule, and all that stuff is captured in that double epithelial layered capsule or cup around the glomerulus, everything starts to go down this proximal convoluted tubule. Now, what happens is after the blood is filtered in the glomerulus, filtration is done. That's basically filtration. You filter a lot. You filter 100+ liters of fluid per day. Okay, and if that starts down this tubule and goes down the first portion, which is called the proximal convoluted tubule, this is where a lot of what we call reabsorption occurs. Now, reabsorption was that second process that we talked about, where we filter a lot, but we take almost all of it back. If we filter 100 liters, we take out back 98.5 liters that's back there, and a lot of that happens in this proximal convoluted tubule, which is the next portion. After the filtrate and after the material, okay, leaves the proximal convoluted tubule, it goes into an area called the loop of Henle, and this is the loop of Henle. It's a loop that will actually dip down way down into the area of the medulla and come up the other way. Okay, it looks like a loop, like a little hairpin loop. Okay, and some concentration of urine occurs in this area of loop of Henle. I'll talk a little bit more about it later on. So we've gone from the glomerulus and the glomerular capillaries capturing the filtrate in Bowman's capsule. That filtrate then starts to go down the proximal convoluted tubule. During that period of time, most is taken back. Now that gets me to a good point. What happens is if you look over here, okay, in this area, here's my efferent arteriole leaving the area of Bowman's capsule, okay, or leaving the glomerulus. And what happens is as soon as it leaves, Look at all these capillaries. There's capillaries all over the place. They're surrounding the tubules. And that's basically for reabsorption and then finally down the road by secretion, putting things back in there. Okay. So all those little capillaries that you see around there are capillaries that extended after the efferent arteriole leaves the glomerulus. Okay. Once that efferent arteriole leaves the glomerulus, it branches off into all these capillaries, and the capillaries surround the tubules so they could pull things back into the from the tubule with the filtrate back into the area of the blood. Okay, so we go through that loop of Henle. Okay, there's a descending loop which is going downward deep into the medulla, and ascending loop that comes up. Okay, once the filtrate leaves that, it goes through the next portion of the tubule, which is called the distal convoluted tubule, and that's this area up in here. This area up in here is the distal convoluted tubule. Again, at this point the urine is being fine-tuned. We're taking a little bit out, putting a little bit back and forth. Okay, and they're trying to make it so that the urine finally gets exactly what it's supposed to be. Finally, once the filtrate gets through the area of the distal convoluted tubule, it comes down what's called the collecting duct. Now the collecting duct, a few things are happening. The fine tuning is occurring in the collecting duct. The last little bit is in and out at the collecting duct, and finally that collecting duct, as it gets wider down in this area down here, eventually empties into the area of the calyces, and there'll be thousands of these that'll empty into the calyx and stuff like that. So basically that's the part. So we start with everything begins at the area of the glomerular capillaries. Okay, and the afferent arteriole goes into the glomerular capillaries. The blood is filtered. All that filtrate gets caught in that area of Bowman's capsule and starts down that proximal convoluted tubule. The blood leaves glomerular capillaries, goes out the efferent arteriole, branches off into what we call peritubular capillaries. Peri means around the tubules, and that's for reabsorption and secretion. Okay, it goes through the proximal convoluted tubule, goes through that loop of Henle, goes through the distal convoluted tubule, and finally gets in that collecting duct and starts to come down that collecting duct, that bighorn, and the urine is just being slight change, fine tuning, just to try to make sure that it's exactly what it should be. Okay, and that's what we have. Okay, so let's look at this a little bit more. This is again the three things that happen. I've mentioned filtration, reabsorption, and tubular secretion. Okay, and those are the three processes in that order. Filtration occurs first. You have to filter before anything happens. Reabsorption means you take most of it back, and then secretion is you put things a little bit back in there. So filtration. Okay, so when we talk about what happens in filtration, fluid from the glomerular capillaries are in that renal corpuscle, and because of the pressure in the capillaries, fluid is filtered from the capillaries into the tubules basically by hydrostatic pressure. Just because, again, the pressure inside the capillaries is high. Why? Because the efferent arteriole is smaller than the afferent arteriole. So that's where filtration occurs in that area of the glomerulus, which would be this area right here. Okay. Finally, what happens is once that we go down that tubule, and actually what they've done is just straighten this out just for diagrammatic purposes. Once they start down that tubule, we end up with what's called tubular resorption. And almost everything is resorbed. Okay, most stuff is taken back, but then we also have what's called selective resorption. Okay, and that means that yeah, I got to bring this back because there's too much out in the tubule, and I got to save that, and it brings it back, and that's called a selective reabsorption. And also they put it back. That the idea is to take the material that it was initially filtered, bring it back, put it in the capillaries, and it could be reused for the rest of the body. Things that they put out but say no, I think that should come back, and that comes back. And again, that's most of the stuff that's filtered. Finally, I get further on down the tubule, okay, and I get to what's called tubular secretion. And a lot of stuff that hasn't been put into the urine so far, like ammonia, uric acid, and stuff like that, is actually now secreted into the tubules closer to the end, just so that what happens is now it gets down the collecting duct and it could go out in the urine and could be eliminated for good. Okay, so we go through filtration, tubular reabsorption, and then tubular secretion. Okay, and that's that particular order. What the urine formation does, it filters out fluids and smaller substances, a lot of fluid and a lot of the smaller substances, and then again reabsorbs the substances that are needed. So it filters out everything, brings a lot of that back. One thing I could say though is think about the capillaries. Capillaries have thin walls, but they have the small little pores, but the pores are really small. Certain large molecules have a difficult time fitting through those pores, and some of those large molecules are proteins. Proteins have a tendency to be large, macro molecules on the size of things, okay? And so therefore what happened is there's minimal amount of protein, almost if any at all, that is filtered out. Why? Simply because the protein molecule is too big to get out through the glomerular capillaries. The holes and the size of the molecule of the protein is just too big to get outside the capillaries, so it stays in the blood. Okay. Glucose is a lot of it is filtered, but most of it is reabsorbed. Most of that glucose is reabsorbed. What happens is when a diabetic patient will show glucose in the urine, and the only reason why they have glucose in the urine is because the glucose levels become so high the tubules can't reabsorb as much. Okay, it's filtered, but most of it's reabsorbed normally. But in the diabetic patients, because there's so much glucose, there's so much reabsorption that they can't get it all back, and some comes out with the urine. So that's how the diabetic patients sometimes they'll go ahead and test the urine to see if there's glucose in the urine, and it's called they say they're spilling urine, and the spilling urine is because the tubules can't reabsorb in a diabetic patient everything that it filters. Why? Simply because there's just too much glucose. Okay, urea, which is a nitrogen product, is partially reabsorbed. Okay, and some also stays in the blood. There's a test that we'll talk about later on. It's called the blood urea nitrogen, and there's always a little bit of this urea in the blood, and that's where they get the test blood urea nitrogen. So when I draw a blood test, one way that they follow kidney function is to actually see what the blood urea nitrogen is. We filter it. Okay, and some of it's reabsorbed, and that part that's reabsorbed gets back in the blood. So therefore, I'm able to detect that urea in the blood and that blood urea nitrogen. If the kidneys aren't working very well, what happens is we have more that stays in the blood. And as a result, the blood urea nitrogen levels continue to rise, which show me the kidneys are starting to fail. Okay, and again, so urea is partially reabsorbed. Some stays in the urine, but most of it's there is going to be some that will still in the blood. And some substances are never reabsorbed. And one thing is creatinine. Now this is important. Creatinine is a breakdown product of proteins, and the only way the body gets rid of creatinine is through the urine. So what happens is the creatinine is very liberally filtered, and it's not reabsorbed. Okay, same thing happens with some drugs. Some drugs, what happens, they filter it all and they don't take it back. They say, I don't want that. It's no good anymore. Just get rid of it. So as a result, some drugs, some substances are filtered, but they're not reabsorbed. If they're filtered, not reabsorbed, what happens to them? They stay in the urine. Things that are reabsorbed get back in the blood. Things that are not reabsorbed stay in the urine. Okay, and that's how urine is formed. And it's a fine tuning of what we get rid of based upon what we need and what we don't need. Okay, and again, this just looks again at the nephron structure and the physiology. And again, if we look up in here, this right here coming in right here. Okay, would be my afferent arteriole, and if you see it does look wider, and these are the glomerular capillaries in here, and this is the efferent arteriole. This right here around the outside is that Bowmans capsule we talked about. So what happens is once that pressure in the glomerular capillaries gets in there, we filter the material and the fluid out. It gets collected in Bowman's capsule and then starts down this area of the proximal convoluted tubule. Okay, so this capillary network with pores and holes and stuff like that in there allows fluids and some smaller substances out. And again, that's by filtration pressure. What do I mean by filtration pressure? There's just so much pressure in those glomerular capillaries that it sort of forces the fluid and as well as the particles out of the capillaries and into that capsule, that Bowman's capsule. Okay, and so that's basically what happens in the glomerulus. And again, this basement membrane around the area of Bowman's, around the area of the glomerular capsule, about the glomerular capillaries, there's a little membrane that also restricts the larger proteins and stuff like that, as well as cells and stuff like that from getting into the urine filtrate. It would be abnormal to see cells in the urine unless they actually come from inside the urinary tract. One of the things that they sometimes find in people with infections, it causes the glomerular capillaries to be leaky, and things larger than they should be that get out in the glomerular filtrate, such as white blood cells, will actually get out. Okay, the capillaries become so dilated and irritated, and the white blood cells, and there's so many of them, they'll get out of the glomerular capillaries and they get in the tubules. And what they do is they stagnate the tubules, and they sort of sit there and they form a large core inside the tubules, and they don't work very well. Okay, they don't move a lot, and as a result, what happens eventually they're passed, and when they look in the urine, they'll actually find like a clump of white blood cells that are all clumped together, and they're called white blood cell casts. And if there's a white blood cell cast, it's indicative of a kidney type of infection. Okay, so that's just looking at about the glomerulus, looking at that afferent arterial, glomerular capillaries, Bowman's capsule, and the beginning of the proximal convoluted tubule. And if you look at the image below, there's a little picture of a Bowman's capsule, what that should look like. Okay, one thing I'm going to point out to you right now is something that's called the Macula Densa and the juxtaglomerular apparatus, which is right next to that, and that's going to come up a little bit later. So remember, that's where the Macula Densa is, okay, which is going to be right in this area. Well, maybe I will. It's going to be right in this area, and the juxtaglomerular apparatus is right down inside here. So just sort of file those in the back of your mind right now. Okay, we're back here. For some reason it blew up, but I think we're back on track now. Anyway, this is going back and looking at Bowman's capsule. And Bowman's capsule is that capsule that's around the outside of the glomerular capillaries. Again, it has a visceral layer which surrounds the capillaries, a membrane that surrounds the capillaries. And it has slits to let certain things pass out into the capsule. It has the capsular space, which is the open space, and it has that parietal layer. And that parietal layer is basically that layer on the outside right here, around the outside, that basically holds everything together. It's the outside of the cup. Okay, so that's sort of, it's important, but I mean, if you're sort of like, you have to know it, that's fine. But it's nice to know, but it's not quite everything I need you to know. So just know that the capillaries are in there. They're surrounded by a membrane. There's a space there that collects the filtrate, and there's a layer of cells around the outside, which are basically simple squamous around the outside to create that cup to hold the fluid in. Okay, and that's what we have, and that's just like a membrane that surrounds Bowman's capsule. Okay, and there we see it again. Let me get rid of the drawing here, the stuff right here. And you see it again. Okay, so the outside, if you look, let's do it this way. If you look out here, here's the outer core, which is the simple squamous around the outside. There's a membrane that surrounds the area of the glomerular capillaries, and then all this stuff in here is the area where the fluid is going to be filtered out, a cup, and then we'll head down through the area of the proximal convoluted tubule. So basically, that's what that really shows. Okay. This is just looking at the blood supply again to the nephron. We talked about how that afferent arteriole goes into the area of the glomerulus, and you can see that pointed by the red arrow. We could see the glomerular capillaries, which is that big ball of capillaries, and we could see the efferent arteriole going out of the glomerulus. Again, by changing the size of these of the afferent and the efferent arteriole, it will change the level of the pressure inside the glomerular capillaries. So if I take that efferent arteriole out here, and if I let me just do it this way, if I decrease the size of my efferent, okay, and increase the size of my afferent, that increases the pressure, because there's more blood that's going to be inside those glomerular capillaries. It sort of gets full. Okay, so by changing the lumen size of the afferent and efferent arterioles helps to regulate the blood flow into the glomerulus. That's why we have an arteriole, a capillary, and an arteriole, which is a very unique way to have our vascular structure anywhere in the body. Okay. Then what happens is, once that blood, like I mentioned before, leaves the efferent arteriole and leaves the area of the glomerulus, we start to form all these little squiggly capillaries around the whole thing, all these little squiggly capillaries, and they're called peritubular capillaries. And basically that's for reabsorption as well as secretion. Okay, so in other words, filtration occurs only at the glomerular capillaries, but what happens is all the reabsorption and secretion occurs in these peritubular capillaries, which actually basically surround the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and the collecting duct. Now let's talk about that proximal convolute tubule. Okay, it's cuboidal epithelium. They have little microvilli, which increases surface area and stuff like that, which helps to increase the reabsorption. You know, that's sort of helpful to figure that out. But again, what I want you to remember is once that filtrate gets in the proximal convolute tubule, everything's filtered that's going to be filtered. This point, the job of the nephron is to take most of that stuff back and get most of it back out. Okay, so reabsorption is what really is the most important function in the area of the proximal convolute tubule. I mentioned that almost all the glucose that is filtered is pulled back in, is reabsorbed. Okay, same thing with amino acids. I want to save the amino acids to be able to make proteins. Okay, water, a lot of that water. Again, if we filter 100 liters, we're going to take back 98.5, and most of that's going to be water. Also, certain ions like sodium, potassium, bicarbonate, chloride, just to try to get it all back, because I'm going to need it. Okay, this happens by active transport or secondary active transport and stuff like that, symporters. But I think I need you to remember that what the major function of the proximal convolute tubule is to reabsorb. Okay, we filtered almost everything we could in the glomerulus, and now in the proximal convolute tubule, we're resorbing. We're getting it back. Most of that gets back. And again, some things like glucose, amino acids, water, various ions such as sodium, potassium, chloride, bicarbonate come back. Okay, because again, filtration is less discriminate. Reabsorption is also just get it all back. Okay. Then we get to the loop of Henle, okay? And that loop of Henle is the area now that's circled by the red. And what happens is this goes deep. There's some nephrons have a really deep loop of Henle that go real deep down into the medulla. As you get deeper and deeper and deeper and deeper and deeper into the medulla, closer to where that renal pelvis would be, the concentration of the extracellular fluid gets more concentrated. And what that does is it starts to suck out water. It starts to suck out water and starts to concentrate the urine just a little bit until we get to the ascending area. When we get up in here, the walls of the ascending loop of Henle are a little bit thicker and they hold water in. Okay, so we to make sure we don't lose too much. Again, we've reabsorbed a lot of our water in the area of the proximal convolute tubule, but as we go down through the loop of Henle, where there's water in it, if I get more concentrated, as I get deeper, as that loop goes deeper into the medulla, what happens? Water goes across that capillary membrane to equalize on the other side, as in osmosis. Okay, we talked about osmosis. What happens is water goes across a semi-permeable membrane to equalize the concentrations. So if it's highly concentrated in here, and it's lower concentrated and more dilute here, where's the water going to go? For the lower diluted areas, say, to the higher, to try to dilute the area that's concentrated. And that's just by simple osmosis. Okay, until we get to this area, like I mentioned up in here, where the walls of the ascending loop of Henle get thick and keep the water in, so we don't lose too much. Okay, now I mentioned here about the juxtaglomerular apparatus. Okay, and that's that area that's right about here. Would be the juxtaglomerular apparatus. I drew it on a previous slide. And the juxtaglomerular apparatus does a couple things. It measures the pressure of the capillary flow in the nephron. Now I talked about this earlier in the previous video. That what happens is when the pressure in the capillaries by the juxtaglomerular apparatus, right by the junction of the afferent and the efferent arterioles, they enter and leave the glomerulus. It tests the level of sodium and water in the arterioles and in the capillary bed. And what they find is if it's low, it means we have to save water. Okay. Also, what it does is also means that what happens is the pressure is low. So in other words, if the pressure going into the area of the glomerular capillaries is low, there's going to be low sodium and there's going to be low water in the blood. So what the body does is at that point there are little granules right next to that juxtaglomerular apparatus that send out that hormone that we talked about in the last video called renin. And then renin turns angiotensinogen to angiotensin 1, angiotensin 1 to turn to angiotensin 2 in the lungs by angiotensin converting enzyme, and that raises the blood pressure, which then solves the problem. So if the blood pressure going into the glomerulus is low, it's tested or detected by that juxtaglomerular apparatus by the macula densa. Renin is secreted. Renin converts the angiotensinogen to angiotensin 1. Angiotensin 1 gets converted to angiotensin 2, and angiotensin 2 causes a vasoconstriction that raises the blood pressure. So the kidneys are very infinitely and intimately associated with controlling blood pressure because of that. Now there's my loop of Henle again, squamous epithelium, reabsorption of water and urea in the descending loop, and that's because the concentration as I get deeper in the medulla, in the extracellular material, extracellular fluid gets higher concentration, so I'm having water go right from the descending loop of Henle into the medulla, okay, and into the area, so it's starting to fill the capillaries down there in that loop of Henle, okay? And then the ascending loop again becomes water impermeable because the wall gets thicker and starts to save water. Again, these are don't remember that they're cuboidal or low columnar epithelium, and again things like sodium, potassium chloride are reabsorbed up in that ascending loop of Henle, the loop going up, and stuff like that. So, but I want you to remember that loop of Henle helps to concentrate the urine, and then finally when it goes up the loop of Henle, you try to keep that urine pretty stable in regards to its level of dilution, okay? Because we can't get water out once it goes up the ascending loop of Henle. And there, again, we talked about that juxtaglomerular apparatus, which is right by, if you look at the loop of Henle, it comes, and when it comes up to right where the Bowmans capsule is, and it goes right by that area called the macula densa, okay? And what happens is the macula densa looks at the pressure in the capillaries, also measures, like I say, the measure of salt and water concentration in that tubule when it's going from the loop of Henle into the distal convoluted tubule. So it monitors the salt and the water in the going from the loop of Henle to the distal convoluted tubule, as well as the pressure in the afferent arterioles and the capillaries. And therefore what happens if that's low, the renin is then produced, and that causes the renin-angiotensin blah 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 blah, and it raises the blood pressure, okay? So all these things are integrated with each other, and that's how the kidney helps to control blood pressure, okay? So let me just go over this again. If there's low pressure measured in the macula densa, okay, particularly with the sodium levels in the tubule as it passes by the macula densa, and as well as being close to the area of the afferent arteriole, okay. If the pressure is low, which is reflected by low sodium in the tubule, okay, low sodium and low water, because where water is, sodium is, and where sodium is, water is. If it's low sodium, there's going to be low water. What happens is then the kidneys release that hormone called renin. Renin causes a conversion of angiotensinogen to angiotensin 1. Angiotensin 1 gets in the circulation, it goes to the lungs, and gets converted to angiotensin 2 by angiotensin converting enzyme, and angiotensin 2, like I said, is a vasoconstrictor. It constricts the blood vessels and then increases the blood pressure. One other thing that happens is that angiotensin 2 then goes to the adrenal gland and causes the adrenal gland to secrete another hormone called aldosterone, and aldosterone goes to the kidneys and says, hey, let's hold back some of that sodium too. Okay, so it holds back some of the sodium, it increases the sodium level in the tubules, and where sodium goes, water goes, so it reabsorbs more fluid, gets more, and as a result what happens is the blood pressure goes up. Okay, so all that, because it increases intravascular volume, so all that makes sense. Okay, the distal convoluted tubule. Now we're getting close to the end. Okay, and this is cuboidal epithelium, and we're talking about here's my distal convoluted tubule out in this area, and the tubules are getting bigger. Why? We're finally getting to the end point and stuff like that. There's some tubular reabsorption, sodium bicarbonate chloride and stuff like that, but what happens in the distal convoluted tubule, the urine's almost there. It's really, really close, and they do a little bit of fine tuning at that distal convoluted tubule. There's going to be not just some reabsorption, but there's going to start to be a little secretion. They're putting stuff back into the urine in that distal convoluted tubule. And again, we get to what's called the collecting duct. And what happens in the collecting duct, a couple things happen, okay? First of all, there's going to be more secretion that's going to be in there, some more reabsorption, but the primary thing that happens is there's a hormone that's put out by the pituitary called ADH, antidiuretic hormone, okay, ADH. If I'm dehydrated and I need to save water, I still have a lot of fluid and a lot of water in my tubule as it goes down the collecting duct. Well, well, the pituitary says, I need more water. I need the kidney to save more water. I want to get it out of the tubule. So what happens is then the pituitary gland secretes this hormone called ADH, and ADH then goes and sucks the water out of the collecting duct and puts it back into the capillaries around the collecting duct to save the water, so the water doesn't go down the collecting duct. Because once that water is down the collecting duct, guess what? It's in the calyces. Once it's in the calyces, it's in the renal pelvis. Once it's in the renal pelvis, it's in the ureter. Once it's in the ureter, it's in the bladder. Once it's in the bladder, it's in the urethra. Once it's in the urethra, it's in the toilet. Okay, so basically that's what the collecting duct largely does. It fine-tunes a lot for water. A few other things are moved in and out, but most of that's been done in the loop of Henle, the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. Okay, so actually, so the ADH allows retention of water if you need to concentrate the urine to save and conserve water, and that's what the collecting duct is largely responsible for. Okay. So what I'm saying here is that what happens in the kidney, it sounds really complicated. It sounds really confusing. It sounds really, and we just touched the surface of that. But what really happens in the kidney is this very precise process of how we make urine. You know, it's just not, it's dumping out and get rid of it. It's not like that at all. We know that what happens is the blood comes in through the afferent arteriole into that glomerular capillaries inside Bowman's capsule and that renal corpuscle. At that point, the blood is filtered. We filter a lot, almost everything, almost indiscriminately. The only things that don't get really filtered aggressively are the large molecules, such as proteins. So we should not see any protein in the urine. We shouldn't see protein at all in the urine. Okay. What happens then is from the efferent arteriole, the blood comes out. It's going to all then turn into and diverge into all these peritubular capillaries. But once that filtrate is made in the glomerular capillaries, it starts down, it's collected in Bowman's capsule around the glomerular capillaries and starts down that proximal convoluted tubule. At this point, the proximal convoluted tubule does a lot of reabsorption. It takes almost all that stuff back. Almost all of it back. Okay, takes a lot of stuff back, again under the direction of what I need and what I don't need, keeping my body in homeostasis with all these chemicals. Then what happens, it goes to the proximal convoluted tubule, it goes to the distal convoluted tubule and does some more reabsorption, and largely in regards to water in that descending loop of the loop of Henle. Then it goes from the loop of Henle, it goes up, it goes past the macula densa. It's measuring again the sodium and the water level in the tubule right where it goes past the macula densa, which is reflective of the pressure inside the capillaries that are in the efferent and afferent arteriole and the glomerular capillaries. And if the pressure is too low, we secrete renin, and that starts that renin-angiotensin angiotensin, blah, 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 like we talked about before. And that raises the blood pressure. So once we pass the macula densa, then we go in the distal convoluted tubule. What happens in the distal convoluted tubule? Basically, I reabsorb a little bit more, but I'm fine-tuning it. I'm not quite as aggressive. The proximal convoluted tubule took a lot of stuff back. What happens is what's left in there that was missed, you know, in the proximal convoluted tubule and the loop of Henle, we take out in the distal convoluted tubule. As well as the distal convoluted tubule starts to throw some things back in that it wants to get rid of. Finally, I end up in what's called the collecting duct. And the collecting duct is that last fine-tuning area for the urine. Again, if I need to conserve a lot of water, the pituitary sends out that hormone called ADH, and it sucks the water out of the collecting duct and puts it in the capillaries around the collecting duct to conserve water. If I'm dehydrated or if I need to save water, that's what happens, and the ADH will do that. ADH, antidiuretic, anti means against, diuresis means urination, hormone, okay, which is basically a hormone because it's in the blood. So it's preventing my urine or limiting the amount of urine by decreasing the amount of water that's in there. The volume becomes less, and the ADH works there at the area of the collecting duct. That's the big thing in the collecting duct. And finally, when I get to the end of the collecting duct, it says, geez, it looks like a big lake out there. And that big lake is the calyx, that minor calyx. The collecting ducts then lead into and dump the urine. It's now done, finished, good, no more adjustments, you're done, dumps that urine into the area of the minor calyx. Minor calyx goes into the major calyx. Major calyx then goes into the renal pelvis down the ureter and into the bladder, blah, 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 and out into the toilet. And that's how urine is made. It's a lot of filtration, a lot of reabsorption, and a little bit of secretion to try to get things and make the urine what leaves exactly what I want to leave. Not too much of something, not too little of something, but just the right amount. Sort of like Goldilocks and the 3 urines or something like that. I don't know. And what happens is it sort of like manufactures the urine according to what my body homeostatically needs or wants. And that's where the kidneys do their magic. When people have a problem, and we'll talk about this in the video 3, when people have a problem in regards to the kidneys starting to fail, all these toxins build up. Initially, it's interesting when people go into renal failure. Okay, at the very beginning of renal failure, they put out tons and tons and tons and tons of urine, volumes of urine. Indiscriminately. Why? Because the nephron stops doing its job. It filters a lot, doesn't reabsorb a lot, everything starts to go haywire. Okay? Eventually, what happens in renal failure, the nephrons stop altogether, and there's no urine function. Okay? There's no urine output at all. There's no urine made at all. And what happens is they look at renal failure as the amount of actually functioning nephrons. The kidney has millions of nephrons. So what happens, they can actually measure the approximate percentage of the nephrons that are working to say how bad the kidney failure is. Once they get to a certain point where they have a very low percentage of the nephrons that are working and the urine is not coming out the same way and we're starting to build up the toxins, that's the time for one of 2 things: dialysis or a renal transplant. Okay, and we'll talk a little bit more about that in the video 3. Hopefully we didn't confuse you too much, because again, you know, this is something we could spend like 5-6 hours on, just talking about the individual idiosyncrasies of what happens more in the proximal convoluted tubule, what happens in the loop of Henle more, what happens in the distal convoluted tubule. We could talk about the transport mechanisms, whether it's active, whether it's assisted active, where it's by transporters, whatever the case may be. There's all different ways, by its diffusion and stuff like that, by osmosis with water, but we don't have enough time for that. So it's much more complicated than this. Hopefully we made it at least simple enough that we could follow it. Hopefully when we get to the WebEx, we'll be able to discuss it so you'll understand it a little bit more than just going through. You'll hear it a second or third time. And but it is a complicated subject. It does take some brain power to think about what's going on. Think about filtration in the glomerulus, reabsorption largely in the proximal convoluted tubule, a little bit less in the loop of Henle, a little bit less in the distal convoluted tubule, and we start to secrete things, putting things back into the urine in the distal convoluted tubule mostly. There's still secretion other places, but a lot of it in the distal convoluted tubule, and then the collecting duct, but the collecting duct does a lot with this ADH and conserving water. Okay, so hopefully that explains it a little bit more. Also explains a little bit about the afferent and the efferent arterioles, why that they're important, how they influence the development of blood pressure or the changing of blood pressure. And we'll see you next for the next video. We'll talk a little bit more about urine and about a couple other things in regards to the kidney. Okay, but this is a lot of the physiology right now in this particular one and this particular video, and hopefully you at least get a little hint of it. Okay, and that's going to be important. Okay, talk to you later. Be safe, be healthy.